Good morning, student. Today I am going to discuss about the first topic, which is introduction and history of microbiology. So before that, what are the learning object we are going to discuss today? So I'm going to discuss about the what is mean by medical microbiology and what are the various branches are there, and what do you mean by clinical microbiology? What is the difference between prokaryotics and eukaryotics, and who are all contributed to the microbiology field, and what are the Nobel Prize they got? So these are the things I'm going to cover today's topic. And what do you mean by medical microbiology? So medical microbiology nothing but it deals the study of the microorganism, either that can be visualized under microscope. So it can be bacteria, virus, parasite, and fungi. So what do you mean by clinical microbiology? Clinical microbiology nothing but whatever microorganism it able to produce a disease called infectious disease, which concern with the prevention, treatment, and diagnosis of the infectious disease. So about this kind of things we are going to discuss about under the clinical microbiology. So there are several categories are there in the clinical microbiology. So let's discuss about the general microbiology and immunology, in hospital infection control, and systemic microbiology. So what do you mean by these terminology will be discussed. What do you mean by general microbiology? General microbiology nothing but, so you will in this general microbiology will be discussed about the general properties of the microorganism and its cell wall and its growth factors, and how the bacteria growing, and what is the growth curve, and taxonomy or etc., which will be discussed under general microbiology. Coming to the immunology nothing but the antigen antibody reaction, which deals the immune system of the immunological mechanism of the disease. And coming to the hospital infection control, so here we are going to discuss about the control measures which prevent the transmission of the hospital acquired infection. So let's have an example of like the COVID, coronavirus, how we are controlling by doing the hand wash technique and proper wearing the mask, like aerosol precautions like that. That can be discussed under hospital infection control. And later the systemic microbiology called in microorganism that infect the various system. So initially olden days of curriculum, which is discussed about the organism wise. Nowadays it is discussed about the organism which involved in the system basis. So that is called the systemic microbiology. So in the systemic microbiology, further it is classified into various clinical syndrome like a bloodstream and cardiovascular infection, and respiratory tract infection, GI tract infection, urinary tract infection. So according to this system, various organisms will involve and produce a disease. So it will system wise will be discussed from this year onwards. And coming to the difference between the eukaryotics and prokaryotics. So microorganism basically classified into eukaryotics and prokaryotics. The except virus which is not comes under this category. So what do you mean by prokaryotics? Prokaryotics the below average of the microorganisms. So eukaryotics having higher order. So this bacteria, blue-green algae comes under prokaryotics. This eukaryotics, like example, fungi, parasites, plants, animal, which all comes under eukaryotics. How they differ, that will be discussed here. So the coming to the nucleus part will be diffuse, and in case of eukaryotics, it will be well defined. The chromosome, in case of prokaryotic, it will be single and one, and that eukaryotic have a many and linear. Because of this we have a nucleus, nucleoprotein, and the cell division will be mitosis. Whereas prokaryotics, in contrast to eukaryotics, it is nuclear membrane will be absent, nucleolus absent, ribonuclear protein will be absent. So these are the about the nucleus. So coming to the other difference between prokaryotics and eukaryotics, the extra chromosomal DNA found in plasmid in case of prokaryotics, found in mitochondria in case of eukaryotics. Coming to the cell membrane, like in case of, I already mentioned that prokaryotics means, example, bacteria, which has a cell wall but does not have any sterol except mycoplasma, which has a sterol. But coming to the eukaryotics, that it contains sterol, some amount of sterol should be there. And coming to the cellular organ like mitochondria, usually it is absent in case of prokaryotics. It is present in case of eukaryotics. Ribosome always 70 S and this will be 80 S. The site of respiration usually the prokaryotic, the site of respiration will be the curved end called a mesosome. The here there the respiratory takes place. But in case of eukaryotics, the mitochondria act as a respiratory center, and pinocytosis nothing but still drinking. So it will be present in case of eukaryotics. It will be absent in case of prokaryotics. So these are the about the introduction of the microbiology. So we have discussed about what we mean by clinical microbiology. That means study of the microorganism by microscope. So we have several organisms like bacteria, virus, fungi, parasites. So these are the study of this microorganism called medical microbiology. What do you mean by clinical microbiology? These organisms able to produce a disease called infectious disease. So by doing the test or by controlling, treating, diagnosing, preventing, by which we can able to control the cure the disease. Dealing with these kind of things known as clinical microbiology. We have several varieties are there, like branches are there: general microbiology, immunology, hospital infection control, and coming to the systemic in microbiology. So now we are moving towards the history. So history, what will be discussed in history? So who are all contributed in the microbiology field and who are all got the Nobel Prize? That we are going to discuss in the history chapter. Okay. So coming to the first scientist, he is the first scientist like Anton Leeuwenhoek. He is the one first identify the microorganism by using the single lens microscope. He named that little animalculae. So just see this thing. This is the microorganisms. Once again. So this is the microscope. So by which he has seen the microorganism, and he named that little animalculae. So he is the one Anthony Leeuwenhoek. Okay. So he is the first one who has identified the microorganism. Coming to the second person. So second person, he is the Edward Jenner. Edward Jenner is known as he is the first person introduced the vaccine. He only introduced the vaccine for the cure of smallpox. He developed a vaccine from the cowpox and treated the smallpox. So why the name came vaccine? So this cowpox, scientifically the variola vaccinia, the vaccine came from the vaccinia. So he is the first person introduced the vaccine. Just remember this thing. Edward Jenner, he is the first person introduced the vaccine for the smallpox. And next we'll move on to the Louis Poisson. He is a father of microbiology because he proved or discovered many things. He is a chemist professor in the France, and he contributed like a principle of fermentation. The bacteria able to ferment sugars, he only proposed or discovered. And second thing, he introduced a sterilization technique by like a autoclave, hot air oven, so by which we can use the instrument in a sterile state. Okay. Next, he also introduced he described the pasteurization of milk. And he also discovered the vaccine for cholera, fowl cholera, anthrax, as well as the rabies by infections. And coming to the other contribution of the Louis Pasteur, he disproved the germ theory of the disease. And he is the first person introduced the liquid culture for isolation of the organism in a pure form. Okay, these are the contribution of the Louis Pasteur. Okay, Louis Pasteur is the father of microbiology. He discovered the fermentation, and he discovered the three vaccine against the anthrax, fowl cholera, as well as rabies. He introduced the sterilization technique and pasteurization of milk, and he's disproved the germ theory. He's the father of microbiology. So coming to the next scientist called Joseph Lister. So Joseph Lister, he is the father of antiseptic surgeon. Why he is the father of antiseptic surgeon? He is the person introduced the dilute carbolic acid to sterilize the instrument and to clean the wound surgery. By doing this, he proved that post-operative infection rate has come down. So that's why he is named as a father of antiseptic surgeon. And next scientist called Robert Koch. He is the most important one for your short notes. Here, he is the German general practitioner. First, he only introduced the agar preparation for the isolation of the pure culture. So, Paul, already we have mentioned that the liquid culture has been introduced. Okay, so he introduced only liquid culture, but Robert Koch introduced the pure culture. Okay, like the solid agar. By using the agar, we can make the liquid media to become a solid, and by which we can isolate the pure culture. And he is the one I introduced the hanging drop method for detection of the motility of the organism. He also discovered the anthrax bacilli, tubercle bacilli, and cholera bacilli. He is only introduced the staining technique. So he told that by doing the staining, we can visualize the microorganism in a proper manner. First, he introduced the aniline dye as a staining agent, and he stained and visualized under microscope. And he said it is a good agent to visualize the organism under microscope. Okay. Next, he introduced two things. One is Koch phenomena and Koch phosphate. Koch phenomena, nothing but the guinea pig previously infected with the tubercle bacilli, it can able to develop hypersensitive reaction when if you introduce the tubercle bacilli and or its proteins. That means if you introduce any agent to that particular animal or patient, if he developed hypersensitivity, that means he is already exposed to that kind of disease. So he proved that thing. That is known as Koch phenomenon. Coming to the Koch phosphate. Koch phosphate, nothing but the infecting agent should fulfill 4 criteria. What are the 4 criteria? So the Robert Koch have made 4 criteria. The microorganism constantly should be present in the lesion of the disease. And second, from that lesion, if you try to isolate, it's able to grow in the pure culture form. And third, the same disease should be developed if you reintroduce to the experimental animal from the isolated organism. And the fourth one, you should be able to re-isolate the same organism from the experimental animal. So these 4 criteria should be fulfilled to become an infectious agent. So this is the 5th criteria, which is not developed by the Robert Koch, but it is added in this infectious agent. Like a 5th criteria, the person should be able to produce the antibody, which should be able to demonstrate in the in vitro method. So the picture shows that first the organism should have constantly should be present. Second, you should be able to isolate in a pure form. Third one, if you reintroduced or from the culture to the experiment animal, the same disease should be produced. And fourth one, it should be able to re-isolate it from the experimental animal. And few organisms which is not able to follow the Koch postulate. So Mycobacterium leprae, Treponema pallidum, which is not able to grow in an artificial media. That's the blood agar mechanica agar. That's why it is not able to demonstrate the Koch postulate. And coming to the Neisseria gonorrhea, the Neisseria gonorrhea, there is no animal model, but it can able to grow. That is the problem. Because of these things, the Koch postulate cannot be able to demonstrate by these organisms. Coming to the Paul Ehrlich. Paul Ehrlich is a father of chemotherapy. He is the first person. He told that the mycobacterium has an acid-fast nature in its cell wall. So he proposed like toxin antitoxin reaction is there. That is called a Ehrlich phenomenon. He is the one proposed the side chain theory for the antibody production, and he is the one introduced the salvarsan for the syphilis. So it is effective medicine for the syphilis. Syphilis is nothing but sexually transmitted disease, which is olden days it was not curable disease, and later he introduced this salvarsan. That's why it is called a magic bullet, and he named it father of chemotherapy. And he identified the Ehrlichia like a bacteria which is belonging to the Rickettsia family, named after him. So it is a Ehrlichia. He got many Nobel prizes and immunology. So also he has he is a founder and director of the Paul Ehrlich Institute in the Germany. Okay, this till now we have covered of the few scientists which major contribution to the microbiology. So in that the Montoni von Leeuwenhoek, he is the first person he is introduced the microscope and identified the microorganism. He named little animalculae. Second, second we have a Edward Jenner. He is the father of he introduced a vaccine for the smallpox by using the cowpox vaccine. By which he has controlled the smallpox, because olden days the smallpox infection was a very huge threat. It was more infectious. It is able to spread from one person to another person with the fluid, and the vesicle will be there, and the patient who's got the infection will be more severe and maybe life-threatening also. So it was the dramatic changes happened because of the Edward Jenner. And Louis Pasteur. Louis Pasteur is the father of microbiology. He has introduced many things, and he has introduced a sterilization technique, and he introduced the vaccines for anthrax, fowl cholera, rabies. So his contribution is major in the microbiology field. And Robert Koch is one scientist. He introduced a Koch postulate and Koch phenomena. So Koch phenomena nothing but we can experimentally we can tell the person is exposed to the infections or not by introducing the antigen or protein to the body. If the persons are developed a hypersensitivity reaction, that means already the patient is exposed to that kind of infection. And second thing, Koch postulate. He introduced the Koch postulate. Koch postulate nothing but it should be the infectious agent should be eligible for 4 criteria. One is the infectious agent should be constantly present in the lesions. Second thing, if you try to isolate, it should get the pure form. Third thing, if you re-inoculate it into the experiment animal, the same disease should be produced. Fourth thing, if you try to isolate from the experiment animal, the same organism should be isolated. These four criteria should be fulfilled to become an infectious agent. And the fifth criteria, always the if something antigen enter in our body, will get an antibody. So that should be demonstrated in the patient's serum. Okay, this is the fifth criteria. And coming to the this is under coach postulate. So this will be essay short notes. Also try to remember this thing. And Paul Ehrlich. Okay, Paul Ehrlich is father of chemotherapy. Why is the father of chemotherapy? He is the first person to introduce the salvarasum for the treatment of syphilis. Syphilis is a major sexually transmitted disease. So olden days it was a not curable disease, but introducing the salvarasum, so the patients able to control the disease formation. So because of that, it is named a magic bullet. So these are the contribution of the few scientists. And so let me discuss other important contributor like Hans Christian Gram. Hans Christian Gram developed a Gram stain and earned Ruska. Okay, he founder of the electron microscope. He is the one introduced the electron microscope. Gram stain nothing but one of the staining technique by which you can able to demonstrate the organism and classify the organism into gram positive as well as gram negative. And coming to the Alexander Fleming, he is the one discovered the penicillin from the fungus, and which is used for the treatment of many diseases till now. And good Poisson is the person introduced the viral culture in the chick embryo. The viruses cannot be grown in the cell-free media, but he introduced the good chick embryo culture, because virus can be cultivated by many methods like egg inoculation, animal inoculation, and cell lines. So he is the person introduced a chick embryo technique. And there are several like Carrie B. Mullis discovered the PCR, and several scientists, so many scientists has been introduced or discovered the many organisms. Let's take an example of Oxton. Oxton identified the Staphylococcus aureus, and Neisser. He is the one introduced the gonorrhea. That's why he named as a Neisseria gonorrhea. And Corynebacterium diphtheria discovered by the Loeffler. That's why the Corynebacterium diphtheria are known as club Loeffler's bacillus. But later only it is renamed in the Corynebacterium diphtheria. And Streptococcus pneumoniae, likewise, Frankel has been discovered. The Clostridium tetani, Kitasato, like this, so many scientists has been contributed in the microbiology field. And this Mycobacterium leprae, already I have mentioned this Mycobacterium leprae just not obey the Koch postulate because of the, it is difficult to cultivate in the culture media. So the Henson has been identified the Mycobacterium leprae. And Yersinia pestis is one of the bacterial infection causing plague. So Yersin and Kitasato has been one of the person behind the discovery of the Yersinia. And likewise Clostridium perfringens. So many scientists have been contributed for the discovery of the organism. So common name and scientific name, for example, Corynebacterium diphtheria. So since the Loeffler identified and club has been demonstrated, it is known as club Loeffler's bacillus. Corynebacterium pseudotuberculosis, Parasis nocardium bacillus. Haemophilus egyptus, Koch weak bacillus. So like that, so many scientific name is there, and common name is each and agent. Mycoplasma pneumonia, otherwise known as each and agents. So these are the multiple choice questions you should remember, and because it will be helpful for your exam. And who all got the Nobel Prize in the field of microbiology? Let's take an example like 1901, Emil A. von Behring. He got the Nobel Prize for the development of antitoxins against the diphtheria. So diphtheria is one of the childhood disease, which is because of this more morbidity and mortality, patients are not able to breathe properly. So he introduced the antitoxins because diphtheria mainly forwarded because of the toxin production. If the toxin production high, the bacteria able to produce the disease. So he introduced the antitoxin. Sir Ronald Ross in 1902, Life Cycle of Malarial Parasites in the Mosquito. So, discovery of positive agent Mycobacterium tuberculosis, the responsible agent for the tuberculosis identified by Robert Koch, which I already discussed in the previous slide. And the malarial parasite is unstained preparation of blood can be demonstrated, that is discovered by Charles Laveran. And anaphylaxis is one of the life-threatening problem. So because of the hypersensitivity, type 1 hypersensitivity, the immediate reaction happen. The patient may go for the angioedema and respiratory distress, that is discovered by the Charles Richard. So like that, so many scientists are there in the microbiology field. And the described ABO blood grouping is there, like a Karl Landsteiner. And already I mentioned Alexander Fleming is the one introduced the penicillin drug for the treatment of the many infectious disease. And poliovirus has been cultivated in the tissue culture by Yender F.C. Robbins. And conjugation by which the bacteria are able to transmit the genetic material from one bacteria to another bacteria, that is proved by leader and Totem. And postulate immunological tolerance, that is Sir M. Burnett. So these are the persons are discovered and got Nobel Prize. And HLA is a major antigen which is decide the whether we can accept the donor or not, because if the HLA antigen is mismatched, so the graft rejection will happen. So that is discovered with the Vanessett and Rossett. So these are the major things happen. Okay. And HIV, discover the HIV, Luck Mondinger and F. Farrell. This is a person, 2008, he have discovered the HIV is one of the major sexually transmitted disease. And this description of the prions, nothing but infectious agent which is not having any nucleus. That is stenyl B protein, which is resistant to many it is resistant to even autoclave temperature like a 120, 115 degrees Celsius. So these are the many contribution. And artemisinin, and these are the anti-malarial drug which is discovered, and parasitic effect of ivermectin in the filariasis, William C. Carpenter. And there are so many contributions are there. So let me discuss about the questions. Okay. What is the correct sentence regarding prokaryotics? Can you anyone able to answer? Prokaryotics, I already mentioned that it is below the average of the eukaryotics. That is whether it has a nucleus, cell membrane does not contain sterol except mycoplasma, cellular organs like mitochondria are present, extra chromosomal DNA is present. I'm just asking the correct answer. You just try to tell which is the correct answer. Yes. So what you said is the correct answer. Like it is cell membrane does not contain sterol except in the mycoplasma. Okay. So this is the answer for this question. Okay. And second, we'll move on to the second question. Who is the father of microbiology? So who's Anthony van Leeuwenhoek is the first person identify the microorganism by using the single microscope, and he named little animalculae. He is not the father of microbiology. Edward Jenner is introduced the vaccine, but it is not the father of microbiology. Joseph Lister, he is introduced he is an antiseptic surgeon. He introduced the carbolic acid for the to control the post-operative infection. So his answer is yes, Louis Pasteur. So this is the correct answer. Louis Pasteur is the correct answer. Coming to the next question, match the correct common name with the scientific name of the bacteria. Correct name. Okay. Latent agents, otherwise known as Mycoplasma, Bacillus, Poachweek Bacillus, Corynebacterium tuberculosis, Club Lophlus Bacillus, which is the correct answer. So here the club Lophus bacilli, otherwise known as Corynebacterium diphtheria. So today we have discussed about introduction. What are the things we are going to discuss under microbiology that has been discussed, and what are the branches are there in the microbiology that has been discussed, and overall contributed in the microbiology field that also discussed today classes. So if you have any doubt, you can ask me. So if you don't have any doubt, so no, just giving 5 minutes to have any doubt, you can clarify. Otherwise, we can end up the session. Thank you.